All right, and we are going to dive into the functional keystone species that are found specifically in your patient's gut.
Yeah. So this is the second favorite part of my test. If you watch our previous videos, the biologically important bacteria are my favorite part of the test because they really tell a tale of what's going on within your microbiome. But over the last several years, I've been speaking a lot about keystone species and how important they are. They are called keystone species because they play a real pinnacle role in not only the structure, but the function of the rest of the microbiome. Many of these species individually are inversely correlated with a number of health conditions. Taking one of the first ones on the list, Akkermansia, for example. So let me share my screen here and just talk through the report just a little bit. So we've identified there's been a dozen or so species that have been designated in a multitude of papers as being keystone, meaning that they have multiple functions. Their levels are directly correlated with health and wellness, and they tend to be the important types of bacteria for us to look at to understand how healthy our microbiome is. So we've listed out these keystone species. We've listed, what they're known to do and why they're important. And of course, if they tend to be outside of the healthy normal range, we provide you the lifestyle and supplementation recommendations for how to bring some of these species up by themselves. So one of the first ones is Akkermansia muciniphila, really important for metabolic health and production of short-chain fatty acids. Akkermansia is a keystone of the keystone species, if you will. What's really interesting is it's the only species in the Akkermansia genus in the gut. There are very few bacteria that are there by themselves with no other cousins in the same genus. Akkermansia is one of the only ones. And Akkermansia is inversely correlated to everything in the metabolic syndrome spectrum or the cardiometabolic syndrome spectrum. There's a total of something like 60 illnesses that it's inversely correlated with, meaning the higher Akkermansia your levels are, the more protected you are against all of those chronic conditions. So here we basically show you the IQR of the average range in the healthy individuals. If you look back at one of the videos we recorded where we go through the details of who these healthy individuals are, these are the median range for the healthy individuals, and then you see where your sample falls in that range, whether you're within the range, well below it, or higher above the range. In most cases, being high above the range is not an actionable thing. It's typically fine. Some people tend to have high levels of certain keystone strains, but the important thing is to have the presence of most of these keystone strains and having adequate levels of each one. And then this one gives you your percentile ranking of where you are in general for the relative abundance of this particular range or this particular species. And then, of course, recommendations if levels are low. So we look at Akkermansia, Fecalum bacterioprosnitzii, which is inversely correlated to a number of inflammatory conditions in the bowel. Rhumnococcus bromai, which is really important for degrading plant-based materials. It's also associated in another study that we did with skin health, an inflammatory response in the skin. And of course, Rhumnococcus flavifaciens, that's another really important keystone strain. So there's a number of them that we identify. In this case, for example, and again, this is a real report, this individual tends to be high in Rhumnococcus bromai, which is good. It's fine. They are higher above the range. So nothing really needs to be done here, but they have no detected Rhumnococcus flavifaciens. So they would want to look at some of these dietary alterations to try to bring up the levels. Now, not detected doesn't necessarily mean that it's not there at all. It could mean also that the levels are so low that we did not pull up enough genetic material to fully sequence it. right? So that's an important thing to understand. If it's not detected, doesn't mean that it's completely absent. It could mean, and it likely means that it's there to some degree, but the levels are so low that we need to work on getting it back up. So if I was this person, I would start looking at some of these types of foods and increasing the intake of some of these types of foods, and prebiotics to try to bring up this particular Keystone strain. But in general, they're doing well because their fecalum bacteria is at a good level. Their Akkermansia is a good level. This strain of Rhumnococcus is a good level. So in general, their Keystone strains seem to be pretty strong, with the exception of one so far. So this is really important because it is very clear from all of the microbiome research over the last 15 years that the adequate presence of Keystone strains is one of the biggest factors in how healthy you are and how healthy and functional your body and your microbiome is. So that's why we really wanted to focus in on the Keystone strains and give you specifics on each of the Keystone strains that have been established as being biologically important. So that's this section. Again, putting this together with the other pictures kind of gives you the big picture representation when you look at the first part of the report, and then you go into the more biologically important bacteria. Then you're coming down, and the biologically important bacteria, remember, are smaller groups of bacteria. The first part of the report is a bigger picture of the ecosystem. Then you go down to the smaller groups. And then here you're going down to individual strains. And these individual strains are critically important. And the good news is there are ways of modulating and increasing these individual strains through diet, lifestyle, and supplementation as well.